Last day of April already. How did that happen? A couple of quick announcements. First one is the DVD on Lambda that some of us watched on Friday should be on reserve in the library. 2-hour checkout. Just go to the reserve desk, the circulation desk to your left, right when you go into the library. Second thing is, I'm sure all of you noticed from carefully reading the syllabus, we have a guest lecture on Wednesday. One of the really nice things about being at Portland State is we're close to another major medical institution, which will remain nameless, and they have a lot of people there who have expertise in flaviviruses, particularly things like yellow fever, dengue, Zika, what's the other one? Forgetting West Nile. So one of those experts will be here on Wednesday talking about flaviviruses, Alec Hirsch, who I think his official association is with the Vaccine and Gene Therapy Institute, which is the West Campus out in Beaverton of that institution, which will remain nameless. Yeah? How long is the whole thing to watch it? Good question. I don't know. We went through most of it on Friday. Probably took us about half an hour. So it's not a, not huge. 2 hours is plenty of time to check it out and watch. And there are various different sections. It's broken up into chapters, and you can look at each of those individual chapters too. Yeah, the overview is really nice. There are a few things also, if people are going to go and take a look at that, which they get into in the DVD, which since we didn't present it in class and I don't expect everyone to be able to see it, are not going to be on any kind of exam. I consider that sort of as a review for looking at various different aspects, which we did discuss here. More sort of Lambda questions and DVD questions. OK, so let's move on here very quickly. If we do have time at the end today, which, knowing you and me and our predilection to go down rabbit holes is unlikely, we may get a chance to talk about, pardon me, TMV at the very end, but I kind of doubt it. And if we haven't talked about it, I'm not planning on asking questions about this on exams. Again, if we do get to it, great. If not, fascinating, wonderful. Go to OSU and take their plant virology classes. So today, we're going to, some people will say, finally start talking about viruses which infect us. And this whole message here, I'm probably one of the very few virologists that call these pico-RNA viruses, because they're small RNA viruses. Almost everybody who works in the field calls them picornaviruses, which I think is completely crazy. But they all call them picornaviruses. So when he says picornaviruses, you say pico-RNA. Yeah, I think it makes it a lot easier to think about. They're small virions, small genomes, and they're RNA. So we will talk about origins, and now that we're finally talking about viruses that infect us, we'll talk a little bit about disease. And polio particularly has been one of the almost huge successes in terms of disease eradication. And then we'll talk about much more interesting stuff, at least as far as I'm concerned, the structure, binding and entry, genome, and then, of course, these are RNA viruses, as I mentioned. So replication versus translation is a really interesting and, of course, difficult problem because these things are always running into each other. And then eventually as they get out. So these pico-RNA viruses are very, very, very widespread. We'll talk mostly about poliovirus, actually for almost the whole rest of the lecture. These are part of what are called the enteroviruses. Enteroviruses infect the gut, which of course is weird when we talk about polio and poliomyelitis. We'll get back to that in a little bit of a second. Rhinoviruses, that's probably what I happen to have collected today. Rhino being nose. This is the most common viral cause for the cold. And we'll talk about a number of other cold viruses, just general sort of sniffles. These rhinoviruses are what causes that for the most part. There are a number of other ones, encephalomyelitis, et cetera. Foot and mouth disease virus. This is a huge problem in domestic animals, particularly cows, goats, et cetera. And many people have had to deal with this in terms of just completely culling huge flocks, herds, etc. And the U.S., I think we're still foot and mouth disease free. And then hepatitis A virus. This is sort of only the first of multiple different hepatitis viruses that we'll be talking about. Hepatitis A is a picoRNA virus. Hepatitis B is a really weird and funky retro-like virus. Hepatitis C, which is the one I forgot to mention about the flaviviruses that Alec will be talking about on Wednesday. There are also massive numbers of these picoRNA viruses in the oceans, and potentially way more of these than there are any of these ones that we care about. But again, the reason we care about these is because a number of them cause diseases, particularly polio, which has been very well studied. Now let's talk a little bit about polio disease. Most people, if they're infected with polio, will never notice, which is kind of weird because a lot of people talk about, polio disease, how nasty it is. Part of the reason, actually, that polio is so nasty is because most people don't notice if they're infected, and they have no symptoms. And so if I have no symptoms, why should I bother with a vaccine or any kind of antivirals? The thing is, being an enterovirus, what does it do? It goes through. And as soon as it goes through, it has a tendency to end up back in an organism. So in very few cases where people even notice a disease at all, you can have paralysis. So 1 in 200 cases. But again, this 200 number is really hard to tell because, again, most people don't even notice that they have been infected with polio. And then of those 1 in 200, 5 to 10% of them actually die. Now, why do people care and why do people actually notice? And that is that most of the people who get poliomyelitis, we'll talk about in just a second, are kids under 5. Probably because they're not quite into the kind of hygiene that many of the rest of us should be. So that's a big deal, and also has to do with the developmental processes whereby polio seems to be a big problem. Why is polio a problem? Again, mostly non-problematic, but in a few cases it is. And that's because it can replicate in neurons, which is not the normal place that it's supposed to be. Again, normally it sits in the gut, goes straight through. But a few times it'll go into neurons, and then you end up with disease. We'll talk a lot more about this Sabin strain later on. This has no replication in neurons because of a change in the iris. What the heck is an iris? Stay tuned. We'll get back to irises in just a second. So poliomyelitis. This is the classic presentation of poliomyelitis, which is here seen in this poor Pakistani child here, but has also been known for literally thousands of years. This is an Egyptian carving of someone who also seems to have this poliomyelitis. What would be a little hard to see here is this person is walking with not really a cane, but something to be able to support them. And so even in those cases where you don't have death, which again is 5 to 10% of the time, you'll have some kind of paralysis. And this is also known as acute flaccid paralysis, or AFP. And so lots of people look at that. It turns out that before vaccination, there were over 35,000 cases of this every year in the US. Now, this was a few years ago, even before I was born. Yes, means really, really old. But the idea there is that, unfortunately, most of us have no idea what polio is like. So I think this also has to do with a lot of vaccines, and we'll continue to talk a lot more about vaccines as time goes by. A lot of this progress, again, in terms of polio, we've now gone to, literally 2 years after the vaccine, there was a 90% reduction, and in 79 it was completely gone. 79, probably most of you weren't born. I was. But we have had very little experience with this disease. On the other hand, it's only been eliminated in most parts of the world much more recently. And we'll see some cases of that a little bit later on. So why does this disease really get eradicated in the US because of vaccines? How did those vaccines start to be developed? One of the really major breakthroughs in terms of making vaccines was the ability to grow cells outside of an organism. The very first vaccines were actually made in animals, which was a real pain, and you couldn't get very much of them. Cell culture was developed in the 1950s. And basically, what people were able to do was to grow these viruses in cell culture. And then the Salk vaccine in 1955 was a massive amount of polio that was actually made, which, you think about it, literally these are humongous vats, tens, in some cases actually hundreds of thousands of liters of active polio that then gets inactivated and then used to vaccinate people. This is the shot, the polio shot, or also known as inactivated polio vaccine. A couple of years later, there was what's called the oral polio vaccine, developed by Sabin. This is the one which is mostly used in a vast majority of the world, but as we'll see, this has a bit of a problem. And the big difference between the oral and injected or inactivated is this whole idea of attenuation. Now, we haven't talked about attenuation at all yet. Everybody know what attenuation is? Yes? No? A few of you? You go down my list and ask. The whole idea of attenuation, this was developed by Louis Pasteur, originally for looking at rabies. And basically, what he had noticed and Jenner had noticed, and we'll talk about smallpox later on, we'll talk about Jenner's vaccination, is that people who had been exposed to disease didn't get sick later, and more importantly, didn't die. So the whole idea here is if you could make large amounts of virus and put it into an animal that would then get sick but not die, you could take that virus, put it back into an animal that would get less sick and not die, and less sick and not die, and less sick and not die. And just repeat this whole process, literally hundreds of times. And then you end up with a virus which can infect but doesn't cause disease. And as we'll see later, this turns out to be a few mutations that have happened in this particular iris sequence. And this was developed by Sabin, and again, this oral polio vaccine. One of the really nice things about this oral polio vaccine, again, is it can be taken orally, doesn't need to be injected. And that means it's really easy to administer. And what kinds of viruses are polio again? Guy, moi, what kinds of viruses are polio? Group of viruses? Ski here. If not, oh. Yeah, I said this is, of course, printed out this list too short. These are new people. So we know. No. Okay. Andrea, also not here. Why did I pick all these ones at the beginning? Andrew. Yeah. So pico RNA viruses, but what kind of pico RNA viruses? What family? What did we talk about right at the beginning? So poliomyelitis causes this neural disease, but where is it usually? Right, enteroviruses. So oral means it's going in the normal route. So that's another one of the reasons that this turns out to be such a nice vaccine. So because of these vaccines, we have done an amazing job at not having to see kids that look like this. And this was great again in the US. By the late 70s, it was gone. However, in the rest of the world, it was by no means gone. And in the mid-20th century, about 500,000 deaths per year. And this is deaths, not just the acute flaccid paralysis. In 1988, there was a start to the eradication campaign at about 35,000 now cases. This is acute flaccid paralysis associated with polio. 2015, 74 cases. 2017, 22. So this is pretty typical with disease eradication. Starts out, you have these great reductions, but then finally finishing things off is really, really challenging. And this number is probably way too low in terms of the numbers of dollars that have been used to try and get rid of polio. There's some great websites here. End Polio Now is one of them, and we'll look at one of the other sites a little bit later on, polioeradication.org. These are really nice because they actually do follow up literally on a weekly basis. So this was April 19th, up to date where all of the polio cases, and this is literally in the world where all of those polio cases are. Notice a couple of things here. Pakistan, and Afghanistan, Nigeria, and then here in the Democratic Republic of Congo, and here in Syria right on the border with Iraq. What is there that's really in common about a lot of these areas? Job, Richie?
No idea.
No idea. Guess.
It's hard to get vaccines to everybody, or they don't want them.
Okay, that's one reason. Anyone listening to the news this morning?
Grindingly poor and war-torn.
War-torn in particular. So there were a number of explosions in Kabul this morning, which is not far away from these sites here. Actually, Kabul is a little bit up here. And a lot of these regions in Pakistan are the regions which are not terribly well under control. What about this part of Syria? Until very recently, ISIS, exactly. And we'll see an example of that just a second. So here, this is actually another interesting case down here in the DRC. These are regions where I don't know if this is where all the rebels are active, but there certainly are around this region here. Parts of Nigeria, Boko Haram was very active in some of these regions. So it's really in places where they're very hard to get to, and there's some serious security issues with that. Now, that being said, this year we've had 8 cases. It's absolutely amazing to me that we know in the whole world that there have been 8 cases of polio this year. That's just, the surveillance that has to go into that is amazing if you think about it. Now, there is something else that I wanted to mention here, and that is that there's also a number of cases. You may notice that there's red and green, unless you're red-green colorblind here. The red dots represent wild polio, and these green dots represent something which is probably hard to read down here, CVDPV. So we'll talk about that in just a second, but this is vaccine-derived poliovirus, which is a issue, and we'll get back to that. But speaking of why there are cases like this, has really to do with this issue. Come on, let's go back a slide. We can go back. That would be nice. Except somehow. So this is why treating for polio is a problem. This is picture Raqqa in Syria of January of this year. It had been about 2.5 years since anybody able to vaccinate in Raqqa for obvious reasons, as we just talked about, and unfortunately just completely destroyed. But this is the classic oral polio vaccine here, where we have, no, my pointer is not working, has to be kept cold and can be treated orally. It's a little hard to tell. This is a little dropper that's going into this poor boy's mouth. And many of these kids are now being vaccinated for the very first time because there hasn't been any access to get people into these areas. This is, again, where there was, I think there were something like 78 cases of poliovirus disease last year. And a lot of that has to do with lack of vaccination being able to be there. So what kinds of polio-derived disease were there? And interestingly enough, even in these places, like in the Syrian-Iraq border, you said that was those green dots. Those green dots are this vaccine-derived polio disease. Okay, this seems like a really horrible thing, right? Why should you be vaccinating people if they can come down with disease? Well, it turns out that, yes, oral polio vaccine, which is the only way that you can end up with this issue, is active polio because it's gone through this whole attenuation process, but is still infectious. And what happens is what happens through that whole attenuation process. Okay, where are we now? Alex Paulo? Is nobody here today? Roya? What happens in the attenuation process? What do you think is happening? From what you know from molecular biology, what's happening to these viruses as they start becoming less and less virulent? Pardon? Yeah, why are they getting less virulent? Why do you think that might be happening? What happens to the virus when you do this attenuation process? Any ideas? Go for it. What do you think is happening? Why would you be less infectious? What happens to viruses? They've got proteins, they've got nucleic acids. What's likely to happen to them? Pardon? They're phosphorylated, they could be modified, but how else might they be modified? What happens if you change a sequence? What would you call that? Nucleic acid sequence. What's that change? What would you call that? Mutation, exactly. So it's mutation that's happened here, but if mutation happens going from one nucleotide to another, what else can happen? Those mutations can revert. They can go back. And so that's exactly what happens here. So the oral poliovirus has a number of mutations, and then those mutations can revert. Happens, however, at very, very low frequency. And these are some data from, I think this was Eradicate Polio, but between 2000 and 2011, there are about 10 billion OPV doses and 580 cases of vaccine-derived polio. So an incredibly small number percentage-wise, 1 in 200 million. If you hadn't had vaccination, there have been 6 million cases. So yes, getting a disease from a vaccine is really a problem, but given the vaccine in general, it's really good. But what's happening now is they're getting more and more of these vaccine-derived because we're getting less and less of the wild-type polio. And we'll see why that turns out to be an issue. And again, there were 74 cases in Syria in 2017, but that was only through September. This year, we haven't had any more. And hopefully, that's something which is going to continue. Yeah, Margot. Okay, so basically, why not? If it's problematic in neurons, why don't you immunize where it might be replicating? Basically, yeah. So the idea is that in general, and this is true actually even for all these vaccine-derived cases, you get mucosal immunity which forms in the gut that protects all of the rest of you, and even all of these vaccine-derived polio cases, they're also in areas where you don't have good vaccination as well, and so that general vaccination, again, through the oral polio vaccine, there's one of the big advantages of oral polio vaccine is it's really cheap. And as I mentioned, it's also really easy to administer. So going intranasally, yeah, you could certainly get it into the nervous system that way, but maybe you'd have things that are replicating there as well. And you also don't want to have potentially this oral polio vaccine that might have mutated, even if it's the wild type, it doesn't get into neurons that well. So you might not want to put it directly next to the neurons in general. Okay, so this is clearly an issue that is going to have to be dealt with. And as I mentioned, this is up to 2011 when we were still getting rid of a lot of polio. Now that most of the circulating polio is actually this oral polio vaccine, now we need to think about changing direction a little bit. Yeah, your question. So the question is, why aren't there more polio cases in the US given, I like to call them pro-disease actually rather than anti-vaccine, but that's a different story. But yeah, it's a great question. And one of the things that in fact happened, and we'll talk about this in just a second now, is because there were still a few cases where OPV actually did lead to active polio. And if you look online, particularly on some of those pro-disease sites, they will mention, oh, this guy got polio vaccine and he came down with polio. No, statistically, it has happened. And so actually what has happened in the US is we've now completely switched over in terms of polio vaccines. It's not oral polio vaccine in the US anymore. It's all a shot, which is inactivated. And the other reason, now even the people are denying doing this, A, there's not oral polio vaccines circulating, and B, we got it low enough that that doesn't seem to be an issue anymore. So, and early enough. So if this whole business had happened 30 years ago, there would be polio cases in the US. So, which might actually, well, again, go down the rabbit hole here, potentially be a good thing because people would see the disease and see how scary it is. But that's, again, another question. So that brings us to, we're having all kinds of trouble here. What's happening now for the rest of the world? What was done in the US was, again, we went back to the inactivated polio vaccine. And we're going to try and start making less and less of this oral polio vaccine. But another advantage of the oral polio vaccine, which I forgot to mention, is because it goes through the gut and ends up in the environment, it can actually serve to vaccinate people who haven't actually gotten the vaccine. So normal transmission of the wild polio, fecal-oral transmission, but this oral polio vaccine can also be transmitted that way. And so there's clearly an advantage to having an actively replicating polio until, as is happening now, you have more cases of the vaccine-derived as you do of the wild type. So hopefully we're getting close to eradicating the disease. A lot of it's now more political than the scientific aspects of things. And of course, we need to do something about these vaccine-derived cases. Yes, Margo. So the question is, for the reversions, when you have mutations and you end up, again, with the attenuation, you end up with mutations, and then reversions. And those, not surprisingly, those genomes have all been sequenced of the vaccine-derived cases. Most of them have had direct reversions. I think there's some examples of slightly different mutations, but for the most part, they all seem to be direct reversions of the mutations which were picked up during the attenuation process. Okay, more questions on polio disease. Yeah?
Why is it that repeated infections with attenuation makes it less virulent overall? Why exactly? Why are the mutations always, or seemingly usually beneficial?
Right, so the question basically is, why does attenuation work? And it doesn't actually work for all viruses, but it certainly does for many of them. And it basically has to do with what we talked about right back at the beginning. A really virulent virus kills off its host. If you're host dependent, is that a good way to survive? No. And so probably this whole process, which is very much an artificial process. You infect an organism, what Pasteur did originally with rabbits, infected them with rabies, took out some of that, put it back into some more rabbits, put it back into some more rabbits, just until the disease level went down. And presumably, again, it's just the case of adaptation, which is happening probably because most viruses actually aren't really that bad, and getting to much more of this mutualist kind of state. The other thing, of course, is this is a completely artificial evolution scenario. You don't hang on to the ones which are the rabbits are dying. You're just getting the virus from the rabbits that are a little bit better. So it's really a selection process, an artificial selection, which is going on there. And now it's almost all done in cell culture, but originally, again, this was done in animals. Okay, more questions about viral disease? We've all got our clickers out because no one else wants to answer questions. Let's see if this will actually work now. Yay! Okay, good. So our first one, again, everybody make their own decision here, and then once we've had a minute and people have decided about this, or if everyone gets the right answer the first time around, we'll move on. what did I say? 90% cutoff is when we. 5. So, 90% cutoff? We'll do 90% cutoff. I'll do 30 seconds so you can talk about this with your neighbors. Go. I'm still aiming for that 100%, so that's me being egotistical. Oh, they're different. So the type 2, there are 3 different kinds of polio I didn't mention before. But yeah, the type 2 are the main circulating ones. And that was in that image that we looked at before. OK, everyone seems happy. We'll vote again, and we'll get 100%, right?
Yeah,
100%. 100%, good. I heard that up front. They told me. I just need to stop it if I see 100%. Last time it was 89% that were correct. So if I do my 90% cutoff, that's my own fault. Okay, are there more than 47 people here? 1, 2, 3. Or 47 clickers, as the case may be. Up, yes, we're over 90%. Yay! Yeah, so I should have mentioned, thank you for pointing out the type 2, which I didn't talk about before. But again, 3 types of polio. 1 and 3 are actually completely gone in the wild. They're not there anymore. 2 is the only one which is still circulating. And that's also the vaccine which is circulating. Yes?
Did the other wild types, were they like more or less intense?
Oh, so the question is, were they worse disease-wise, 1 and 3? They just seem to be less prevalent, and I don't think anyone's got a really good handle on why or why not, as the case may be. Yeah, Jacob. Oh, are there 1s and 3s that are still available in labs? Yeah, we'll get back to that a little bit later on. But yes, the answer is yes. But you can also just make it. So.
And the shot that you get now, that's just for type 2, or does it also
Oh, the question for inactivated. That's a good question. I don't know, but I should find out. It definitely has 2. The question was, does the IPV have 1 and 3 as well, or just 2? And the answer is, I don't know. I should, and I will find out. OK, yes, so the answer is B. Now, we finally get to talk about the interesting stuff. I forget this disease business, having to do with the structure, replication, et cetera, of these different viruses. The structure of polio is a pseudo T equals 3. Oh, man, I need to print these out bigger. This pronunciations of these names, I should ask people earlier. Or print them out bigger, as the case may be. Athalale, Adam? No? Victoria Hall? Yeah. What's the question? What does pseudo T equals 3 mean?
It's almost like a T equals 3.
Why is it almost like a T equals 3? extra credit. Your college is giving me extra credit for that. So what's that pseudo? So if it's a T equals 3, what does that mean? Getting in between the pentamers. It's also then quasi-equivalent. So what is the quasi-equivalent? Also about equivalent, yes. But what else does that mean when we talk about a quasi-equivalent structure? Okay, ask your neighbors, what do they think? Different proteins, so that's what the pseudo is about. So quasi is that you've got 5s and 6s. The pseudos, you have different proteins. So lo and behold, here we have VP1, VP2, and VP3, conveniently here in different colors. Not that they're in different colors in the actual structure. So blue, green, and red. But in 5-fold symmetry here, hexamer here, 5-fold symmetry here. And because of the fact that it's a T equals 3, even if it's pseudo, how many capsid proteins is it going to have? 180, exactly. So 60 each, conveniently. So yeah, because of that's the way the structure is. If you just think about the soccer ball, and I brought my T equals 3 icosahedra here, so you can take a look at that. That's for the external proteins. Turns out there's also a VP4, again, ridiculously creative naming of all of these things. That's sitting on the inside, and there are also 60 copies of that. Usually, if you think about where virions attach to cells, they're going to attach at the 5-fold axis, because those are the things that are sticking out. Well, interestingly enough, in polio, they don't. They actually interact around the 5-fold axis in what are called these canyons between where all the subunits are sticking out. And that's actually a little bit easier to see in this image. Here is your 5-fold axis and these canyons all around that 5-fold axis of symmetry right here. That's where the receptor binds. But if we look now at the individual proteins, VP1, VP3, and VP2, obviously they have exactly the same structure, right? You believe what Stedman says. It's this beta barrel structure. So we talked about beta barrels already. The double beta barrel structure, which is usually where they're sticking up relative to the surface of your virion. That's you see in STIV, in all of these structures from the viruses from all different parts of life. Here, now this beta barrel is sitting down flat on top of the structure. And so here's the beta barrel, which is present actually in VP1, VP3, and VP2. But it's now a flat structure, and all of these fit together in order to make your pseudo T equals 3 structure. So that's the overall structure that you have of the virion. Now, what's the next thing that has to happen? You've got to get in. That's this process. Again, we have interaction with receptors in this canyon on the outside of the 5-fold axis of symmetry. VP1 is here at this 5-fold axis of symmetry. The genome's now on the inside. The virion's up here. Here's the cytoplasm. What happens? What happens here is actually really similar to what happens with a lot of bacteriophage. There's a conformational change in an internal protein, which then makes a hole in the membrane. Makes a hole in the membrane. The genome is basically posted out through that hole and brought inside the cell. Now, you'll notice a couple of things about the genome. There's this VPG thing sticking at the end, the gray sphere. This is a protein that's attached to the end of the genome. And it does seem to be very important for this genome entry. If you don't have a protein associated here, you don't seem to get genome entry that takes place. And that VP4 that we mentioned was on the inside. That's what seems to be making this pore structure. So again, everting and going into the inside of the cell. Very similar to what you see with something like bacteriophage T7. Core structure makes a hole. Phi-X174H makes a hole, etc. So that's the entry process. Once this genome comes in, positive strand RNA will end up being translated. We'll take a look at that in just a second. But first, wanted to mention the poliovirus receptor. Poliovirus receptor, also known as CD155. I wouldn't remember that, but PVR, very conveniently named because it's the poliovirus receptor. Has immunoglobulin-like domains. And who are we up to now? Nathaniel Brown? the question is why immunoglobulin-like proteins are commonly receptors. They're on pretty much every cell, but what else about the structure, which is potentially a reason that they're used for many receptors? What do immunoglobulins do? They bind to things. And viruses have figured out evolutionarily that they like to bind to things. So if you're trying to get into a cell, have something that binds to things. So pretty typical. There are other ones for others of these picoRNA viruses. Many of them include immunoglobulins, but not all of them. And so many different things here, aminoglycans, integrins, et cetera. But the majority of them are going to have these immunoglobulin domains, again, which like to bind to things. And not surprisingly, again, viruses have evolved to deal with these particular situations. So what comes in with this genome? This is the genome of poliovirus. And when we talk about RNA virus genomes, we're going to be very interested in two particular regions of the genome. And whenever we talk about a nucleic acid, what are the two most important regions? 5' end and 3' end. So 5' end of the poliovirus genome has this protein attached to it, virus protein G attached to the 5' end. The 3' end has a poly A tail. So poly A tails, as we've talked about before, unlike the cucumber mosaic virus, which has this funky RNA-like structure at the end, here we've got a normal poly A tail, great for translation, great for protecting the end of your genome. So, and as we'll see, for the various different RNA viruses, they're going to have different things at either their 5' end or their 3' end. VPG is very interesting. Again, it's covalently bound to the end of the genome. But what does that mean about translation? Okay, who's up next? Where are we? Other Adam. Adam. Yeah, what do you mean? What does having a protein bound at the 3' end of your genome mean about translation?
You're going to have to get rid of that protein in order to translate.
You have to get rid of that protein in order to translate or have a different mechanism, which we'll get to in just a second here. So if you look at the rest of the genome, one of the first surprises when this was sequenced by actually Vincent Racaniello, those of you who listened to TWIV, this was part of his, I think his PhD work when he was in David Baltimore's lab, was determining the sequence of this genome. One of the really strange things they found was it was all one open reading frame. Literally one start codon, one stop codon. A very, very, very large open reading frame making multiple different proteins. And this later was found to be a very common process called a polyprotein. So multiple proteins all just hooked up C-terminus to N-terminus, C-terminus to N-terminus, C-terminus to N-terminus. And as we'll see later, these that are here at the 3' end of the genome are your structural proteins. At the 5' end of the genome are the non-structural proteins. And this is pretty common for these virus genomes that have polyproteins, and as we'll hear on Wednesday, very common there as well. There also are non-coding regions, 5' end and 3' end, which usually turn out to be very important for regulatory purposes, for all RNA viruses. And as we'll see here, this is where you deal with the fact that you have a protein bound in the end of your genome instead of a nice cap structure, and also for replication purposes. So let's talk a little bit about how these viruses deal with the fact that they have a protein bound to one end. Normally, you have a cap, 5' methyl cap, which of course in the plant viruses is generated by what, Gino? Yeah. What generates the cap structure in plant viruses? You can make a guess. Somebody found it up front. So they found it. It must mean they've been in their notes. caps are, again, are really important for translation. So what's a cap made of? What's the really specific thing that happens in caps? You have a covalent modification to your RNA. What's that modification? Methylation, exactly. So there's a methylase in the viral genome that does that, because all of this is happening at a, as the virus comes into the genome. So why methylated? Because that's what this cap binding complex interacts with. EF4G, EF4E, ridiculous alphabet soup. Again, eukaryotic initiation factors for translation. The one that we're really interested in here is EAF4G. But EAF4G interacts with the cap structure and then brings all of the rest of the small segment of the ribosome, so you can have EAF2 and the initiator tRNA. This will chug along your RNA until you get to the AUG, and then you get translation. So this is normal cellular process and exactly what happens in those plant RNA viruses. It's a cap made by the viral proteins, and we have this normal process. Well, what about these genomes that have proteins bound to the end? Well, they use these irises. So what's an iris? Again, acronym, a little bit more alphabet soup. Internal ribosome entry site. And basically it is exactly what it says it is. So internal ribosome entry site means you don't need a cap. And so you can get cap-independent translation. Now, it's cap-independent, but it's not independent of all of the initiation factors. So particularly, you need EAF2 and usually EIF4A. What you don't need is EIF4G. And we'll see that EIF4G actually gets chopped up by viral proteases. And why do we think this might be a good thing? Who's up next? Ali? Daniel? Not David? So why would it be to the virus's advantage to cleave one of these general translation factors? Exactly. So it blocks translation of cellular machinery, which means you get a lot of translation of viral proteins. Hey, perfect as far as yeah. So we'll get to that in just a second, but they are made as part of that polyprotein. So at the very beginning, you haven't made these proteins, which is good because you're going to be getting translation. And then later on, I think we actually even have an image of this, where you can see that almost all viral proteins are made, but at the beginning you see all the normal cellular proteins that are made. Yeah, exactly. We'll get to that in just a second. So, yeah, so just quickly looking at some of these irises, there are lots of different internal ribosome entry sites. This is just the one that you see in polio and polio-like viruses. It's all about RNA secondary structure, which should sound really familiar based on those RNA phage that we talked about before in terms of regulating translation. They all have these pyrimidine-rich sequences. Pyrimidines are, of course, the little ones, long name, short, small nucleotide. And then these other sequences that are usually present in each of these irises. And we'll talk about this particular structure here a little bit later on as well. All of these RNA viruses have very specific secondary structures. And those secondary structures are absolutely critical for virus replication. And we'll talk more about that as we move along here. So getting back to your comment about polyproteins. So these are made, as I mentioned, as a single RNA. They get translated not from a cap, because there's no cap, But from this iris, you translate as one ginormous protein. And I think in the case of polio, it's about 1,000 amino acids, or maybe even more than that. So then inside this polyprotein, you have a number of sites where we have internal proteolysis. And what's really bizarre is that these proteases are actually active even as they're made as part of a polyprotein. So they can literally cut themselves out of this protein and then go and cut other bits of the rest of the protein. So here we have this is actually 2A is a separate protease which is made in these genomes. Most of it is this 3C protease. This 3C protease is also the one which chops EIF4G, so later on in the process. So one big protein chopped into smaller pieces, and then eventually is going to end up with all of these individual pieces, with this one exception right here that we'll talk about, well, at this point probably on Friday, as the case may be. So this is the process. We have all these proteases chopping up the genome. Okay, more questions about this? We can get 100% on this next clicker question. Absolutely, good. I like to hear that. So, minute to go. If we can actually start it, there we go. You always put them in caps, right? Okay, is this 90% or not? Let's see. 88%. I'm going to leave it at 88% because I want to move on here. But yes, it's the viral proteases. Not going to give you a chance to talk about it. Sorry, because we're getting a little behind. So my bad here. So again, if we go to 90% all the time, we may be here all week, which I'm sure some of you might prefer. But okay, I did want to talk about genome replication here briefly and make sure we get through this today. Genome replication of these viruses is just like we've talked about with the RNA bacteriophage, always going to be a problem, because if you're translating a genome, replicating it is going to be a bit of an issue. So how does this happen? Turns out that these viral proteins, again, they all get chopped up in individual little pieces. One of those little pieces, as I should go back and maybe look at this here, if this would work, it's not going anywhere, is the VPG protein. So that VPG protein is made, again, as part of this polyprotein, gets chopped apart. That then goes and finds membrane vesicles. And we'll see that membrane vesicles are really important for virus replication of pretty much all of the viruses we'll talk about for the rest of the course. They help to concentrate proteins, and then, of course, if you're an enveloped virus, you're going to be picking up the envelope from there. These polioviruses are not. So VPG associates with these membrane vesicles, and it has two U's that are added to VPG. These two U residues are complementary to this sequence in the middle of the genome. The name is not really important. Now we have VPG with two U's attached to it. U's love to base pair with A's. So this will now bind to the end of the genome, and now your polymerase will polymerize all the way down the genome until it gets to the other end. This is now your negative strand genome. Negative strands don't have irises, so they're never going to get translated. So now you can make lots and lots of copies of your positive strand. How do you make sure that you're not getting translation here when you're trying to make this minus strand? The way this happens is you have a secondary structure that I pointed out when we were talking about the irises that's even further towards the 5' end of your genome that your polymerase binds to, and also a cellular binding protein. It's a poly-C binding protein. Turns out to be important for a lot of these RNA viruses as well. Cellular proteins, viral proteins, which will block translation here. Once you block translation, now you can get replication of your genome. Once you have a couple of negative strands, you're never going to translate these because you don't have iris sequences. You can make many, many copies of your positive strand. So what makes these RNAs? It's a RNA-dependent RNA polymerase, also known as the 3D polymerase. I love the three-dimensional structure of the 3D polymerase. And as all of you recognize immediately, what kind of fold does it have? It looks like a DNA polymerase, even though it's an RNA-dependent RNA polymerase. So we talked about the T7 DNA-dependent RNA polymerase. It has a DNA polymerase-like fold. All of these RNA-dependent RNA polymerases also have these DNA polymerase-like folds. So T7 and all of the RNA-dependent RNA polymerases that we'll talk about for the rest of the class all have these DNA polymerase-like folds. And what that means is probably DNA polymerases actually came later. They started out as RNA-dependent RNA polymerases and then eventually figured out how to deal with DNA. So yeah.
T7 was a single structure, if I remember correctly?
Yes, exactly.
Is this a single
Yes, also a single protein. So it's not a multimer and has the classic, again, supposed to be right hand-like structure that grabs the template right in the middle here. And right in the palm is where you have the active site, which are the metal residues. Yes? Pardon? Yeah, Flint is the other textbook in the library, by the way. Yeah. So that's why when I put up these references, they're all to that. Their pictures are much nicer. Also, the book costs about 3 times as much as our textbook, which is another reason it's in the library and you don't all have it. OK, so that's replication. The next step that we need is assembly. How do these virions get put together? Again, they're all made as a polyprotein. So you end up with this one ginormous protein that gets chopped into little pieces, particularly by the 3C protease. But instead of making VP1, VP3, VP2, and VP4, it actually gets put together as this VP0 precursor. And we'll see what happens to that in just a second. So you have these that get put together as trimers. Those trimers assemble into pentamers. This should sound really, really familiar because it's very similar to the way that Phi-X174 gets put together. And then you put pentamers together. These pentamers together pick up the RNA. Exactly how they pick up RNA is not entirely clear. There's not a nice portal that the RNA gets stuffed into. Do they get assembled around the RNA? Again, this is really still pretty controversial how that happens. But then you'll have this provirion, and virion means that it's got nucleic acid in it, but provirion means it's not quite done yet. What happens? You have one more proteolysis step that takes place. And that last proteolysis will now give you VP2 and VP4, which is what you have in the final virion. So why have all of these steps of proteolysis? Okay, A, you've got your polyprotein, you need to chop into other pieces. But if you think about virions, virions need to be metastable, right? So they've got to be stable while they're being made and when they're on the outside of the cell. But as soon as they interact with receptors, they've got to be able to release their genome. So how can you get metastability? Very often, this is also going to be through proteolytic cleavage. So we talked about proteolytic cleavage for receptor binding proteins, for flu, HA, etc. Now we have proteolysis, which is involved in keeping everything stable until it gets out of the, almost ready to go outside the cell, and then get broken down. And you remember VP4 is the one which is helping to bring VPG in the genome in. So if it's still stuck with VP2, you can't have that conformational change. It's not going to undergo its infection process. So that's how the assembly process takes place. And of course, you also need proteolysis to break up your polyprotein. What else do you need proteolysis for? Shutting down cellular translation, which is what we can see here. As I mentioned, EF4G gets cut by the viral protease, and you go from all the cells being, I'll say all the proteins being made to just viral proteins being made over the infection process. This here is using radio-labeled amino acids. If you include radio-labeled amino acids right after infection, beginning, you're making all kinds of different proteins, and as time goes by, you end up making only basically the viral proteins, which are already cleaved into these individual pieces because they're still being made and that protease is still active because the protease has broken things down. If you just look at general protein synthesis, as the cells would normally be growing, protein synthesis goes up. Virus infected, that protein synthesis goes way down, and then you start to see all of these viral proteins that are being made. Turns out that the 3C protease does not only cleave the translation proteins, it also cleaves part of the RNA polymerase. Why would you want to cut RNA polymerase? Just like the reason you want to cut EAF4G, because it's a cellular process that you don't need because all the RNA is coming from your RNA genomes. So you block down cellular translation, you block cellular transcription, so you end up making all of your virus genome. And the last thing that they do here is they block transport of vesicles. Why are vesicle transport being blocked? Because you want to have lots of vesicles to be able to do your replication process, because that's what VPG binds to in order to provide your primer for the rest of replication. So one thing that I forgot to mention, and we'll do now just right at the end of class, is what do you think is happening with VPG? You notice it's covalently attached to the genome. What does that mean about the amino acid sequences that you have in VPG? One particular part of the VPG sequence. It must have a OH that is being extended from. And it turns out it's a tyrosine, and we'll see tyrosines being attached to the ends of genomes many, many times. Yeah, you had one more question here. We'll finish up.
Yeah, I don't see a lysis protein on the genome. So if they are released through lysis, right? How does that work?
Basically, how do they get outside of the cells? We've talked about lysis proteins, particularly for bacteria so far. Eukaryotic cells that these guys infect are pretty darn wimpy, and so it turns out just assembling enough of these, they will eventually burst out through the rest of the cell. So they don't seem to require any particular kind of lysis protein. So, why do we need to not worry about polio anymore? Maybe we should. Turns out you can make polio in cell-free extract, and you just need to order the sequences, and it works. So there's an interesting question about biodefense and what can we do now in our basements. And Jacob, don't think too hard about it. So with that, again, on Wednesday we'll have Dr. Hirsch. Hopefully we'll get everything recorded there as well, and we'll see you then.